And they're inhibitors, so like how they work, like the mechanism, and inhibitors of enzymes. So you can kind of manipulate their function to your advantage. Like if there's a pathway that you want to shut down a little bit for whatever medical reason, then you can use inhibitors to do that. And then there's natural inhibition in your cell, right? Like if you don't need to do a certain reaction, then you can shut down that enzyme pathway. And what are enzymes doing? Like what is their mechanism? Speed up metabolism and chemical reactions, yeah. I'm just going to abbreviate it, reaction. And then I hear the word metabolism. So metabolism was anabolism and catabolism. And then we did talk about what is a chemical reaction. It's basically that like atoms collide with each other and they either form or break bonds. So describe to me so that I could draw it what an anabolic reaction would look like in general. What's happening? I'm building up a molecule, right? Ana means up. So that would mean that I'm taking some parts that are separate and I'm putting them together. Okay? And what does it require for me to do that? Energy, which also known as ATP. And we just saw several different diagrams that just said the same thing kind of repeatedly. And then what's catabolism? It's the reverse thing, right? So I'm breaking it down. So I'm going to take my big thing that I have, and I'm going to break these bonds apart so that I'm going to have the separate parts. And then plus, I get energy from doing that. So it's a little abstract because you're like, I can't see the energy. It's in the bonds, I guess is how we'll just think of it. I'm kind of oversimplifying. It's like complicated. So when you take in a food molecule, the energy is like hard to get to. It's kind of like dispersed throughout it. So you convert it into this usable form, which is ATP. The whole point of metabolism is it's part of like a dynamic equilibrium where it's not that they're equal to each other, but you're always trying to make more energy than you're using up. Like that's how you stay alive. And you need energy to do work. And work is like whatever it is you need to do to stay alive. So you need energy to do work, because you remember that we said all living organisms require energy. And as far as we know, living organisms have an ATP-based metabolism. And that's one of the reasons why viruses, we're not sure if they're alive because they don't really have metabolism. Like they don't require energy. They just use your energy to copy themselves. So they're not really able to do it themselves. So that's what we talked about was what is metabolism, which it's anabolism and catabolism. It's all the chemical reactions in the body. Some of them generate energy, some of them require energy. It's sort of a balancing act, but you always want to be making more energy than you're using up, essentially. Nothing really gets lost. Like it's one of the laws of thermodynamics. Energy is not created or destroyed. So all the energy that's already in existence just gets transferred into different forms. And so you're transforming this chemical energy in the bonds into more usable energy. So that usable energy is called ATP. ATP is an acronym which stands for adenosine triphosphate. So they call this the energy currency of the cell. So the way that I think about it is like, let's say you had to pay for some kind of service. Back in the day, like the 90s, if you needed money, like most of the time, you had to go withdraw it, like from the bank. Like, and people actually wanted to go to the bank, I guess. So you go to the bank and you take out your money, or you write a check. Like, which then it's like, oh man, I forgot I wrote a check and now my account is negative or whatever. Like it's less convenient to do it that way. So now like you can spend money like crazy. Like Amazon one click, yeah, like go like you just pay for your stuff like immediately. And to me, like that's kind of the difference in the energy. Like when you eat food, it has energy, it has the potential for energy, but it's not immediately accessible. So imagine that like your money is held at a bank or like several different banks. Now, if you have a debit card, or even like the electronic methods we have now, like Facebook Pay and PayPal and stuff, you can utilize your funds much quicker to pay for the work that you need to have done, the goods that you're doing. And so ATP is like the debit card, or like the electronic payment. It allows you to get to the energy more easily. So again, it's kind of abstract, like the energy is stored within the ATP molecule. And so this is its basic structure, which I like this diagram because it's like very simple. It has a sugar. Remember, anytime you see an ose, it's a sugar. So it has a sugar that's called ribose. It's got 5 carbons on it. So there's a carbon, it's a carbon ring. And then it has this compound that's called adenosine. And adenosine is kind of similar to a nucleic acid. It's got like a nitrogen, a hydrogen, and some other stuff hanging on it. And then the reason it's called triphosphate is because there's 3 phosphates. So the last phosphate, it has the highest energy bond. And so it's the removal of that 3rd phosphate that releases the energy that you need to do the work. So your body constantly cycles back and forth between ATP and ADP, which would be adenosine diphosphate. There's also another slightly lower energy molecule that we don't talk about, which is called GTP. And it's the same kind of concept, though. It's an energy storage molecule. But we're not doing cell bio, so we just talk about ATP. So essentially what happens is you already have ADP like in the cell. Like that's something that you just naturally produce. And then you also have inorganic phosphate hanging around through diet and just breakdown of other things in your body. So then you get energy from sunlight if you're a plant, or you get it from chemical, if you're food, you get it from chemical food if you're like a human. And then what you'll do is, and this is the weird part that I don't talk about, like, 'cause people are like, well, how does the energy get on there? I'm like, I don't know. Like, just imagine that it transfers from one form to another. And so the energy that was in the food or the sunlight gets transferred essentially onto that 3rd phosphate bond. So you go from ADP to ATP, and ATP is the higher energy storage of that energy so that it'll be ready when you need to do work. So then let's say that you need to do some work. I got to move this protein from point A to point B. I need to fix my plasma membrane. I got a little tear in it. Whatever work needs to be done, now that ATP is harnessing the energy that you can use to do that work, and so you just release it by removing that 3rd phosphate. When you remove it, it's called dephosphorylation, and when you put it back on, it's called phosphorylation. So your cells just go back and forth between doing that. So that's ATP. It's a storage molecule, I guess, to store energy that is needed to do work. And it's stored in the chemistry of the bonds, like how they're together. All right, so this is sort of related. Like the types of reactions that occur in metabolism are typically what are called redox reactions, which if you had chemistry, you've heard this term before. These things are kind of happening at the same time, so when one thing gets oxidized, the other thing gets reduced. We're not in here, again, balancing any equations or anything, but it's just terms that we have to know. So oxidation is the loss of an electron. So the way that molecules can bind to each other, there's different kinds of bonds that they can have. Like they can share electrons, or they can have it where one side actually has more of the electrons than the other. Electrons are negatively charged subatomic particles. So in the middle of the atom, you have the nucleus that has the neutrons and the protons, and the electrons are circulating around the outside, kind of in a cloud. And that's how the bonds form. It's the interaction between the electrons. So the outer shell wants to have a certain number of electrons, like naturally. But if you strip those off, then that is called oxidation. And when you try to pull that electron off, it actually releases energy, because that atom doesn't want to let go of the electron. It's like trying to hold on to it. So when you pull it off, it releases the energy that was holding it like on there. So when you lose an electron, that's called oxidation. And those are also known as dehydrogenation reactions, because usually you're going to lose that electron as a hydrogen. And the reason that is because hydrogen only has one electron. You're not going to separate the electron from the hydrogen, you're just going to pull the whole hydrogen off. So you'll often see hydrogen, like in chemistry, they'll kind of denote it like this, because it carries its electron with it. So if it was bonded to another carbon, this whole thing would just come off. So if that whole thing comes off, then that's called oxidation. Okay, so you're just going to pull that thing off of whatever you're breaking down. So essentially, when you do catabolism, you're oxidizing your food. Like you are ripping off electrons to release the energy until there's kind of not really anything left. So you're putting it down to the bones. Now reduction is where you gain an electron. So imagine the opposite thing. Imagine that there wasn't an attachment here and you're trying to put the hydrogen back on. That would be the reduction part. The reason it's called reduction is because you are reducing the overall charge. Because electrons have a charge of -1. So if you add an electron, you're reducing the overall charge. So if the charge was like +2, when you add an electron, you're going to reduce it to +1 because you're adding a negative charge. So that's how one of the ways that I remember the difference between the two. Reduction is reducing the overall charge, and that's because you are gaining an electron which has a more negative charge. So there's different acronyms that you can know to remember this. The one I learned in school was LEO, the line says GER. So LEO is loss of electrons, oxidation. And GER is gain electrons, reduction. There's also oil rig. I don't know if people have learned that one before. Oil rig is oxidation is loss. And then rig is reduction is gain. So there's other, you can make up whatever you want, okay, like to remember it. Visually, this is a simple representation of what's happening. During the oxidation stage, remember oxidation is loss of electron. So in order to lose an electron, that essentially means you're donating. So the donor of the electron becomes oxidized because they lost the electron. Now at the same time, there are things that are electron acceptors. They're ready to catch an electron. And so the electron that came from the donor is going to get picked up by the acceptor. And now that they've accepted the electron, they are reduced. Because again, by having that additional -1, they have a reduction in their overall charge. So now, hopefully you can see then, can't this oxidized donor now become an acceptor? Because now it has a space to accept one. And then can't this one become a donor? Because it now has an electron, it could donate it to someone else. And so that is what essentially is happening in the electron transport chain. That process of passing the electron back and forth down the chain is called oxidative phosphorylation. It's a set of redox reactions like this that eventually is going to cause rapid phosphorylation of ADP into ATP. And so you're going to end up making a bunch of ATP when you get to that step. And we'll go over it again, but that's why it's called oxidative phosphorylation. Because it's a set of reactions like this, redox reactions, where you're passing that electron down the gradient, which causes a separation in charge of the positive and negative. And then it's going to force those positively charged hydrogens into the enzyme that's actually going to make the ATP. So it's like a little molecular machine, essentially. Okay, so a lot of metabolic reactions are redox reactions. Okay, so I mentioned before that we were going to talk mostly about carbohydrate catabolism because most things need a carbon source in the form of sugar. That's a major energy source. So for humans, it's typically glucose is our main sugar. We're trying to get to the glucose. So if the glucose is in another molecule like fructose or sucrose, we have enzymes that will break it down so that we get to the glucose, and then we can do glycolysis, Krebs cycle, and oxidative phosphorylation, also known as the electron transport chain. So as you go through this, the amount of energy becomes more, and that's because at first it actually costs you energy to get glycolysis started, and so your net energy is lower, but you're still going to get some energy. So most living things can do glycolysis. Like that's like a basic of life that you're able to just make a little bit of energy. And then the two other steps are what's called cellular respiration. And so in humans, that's the point at which we give off CO2 as our byproduct. And the reason that we have to have oxygen is that we need it to finish the electron transport chain. Now, some people have had a misconception that in order to do metabolism, you need oxygen. You don't, because there's organisms that are anaerobic. They don't use oxygen as the final electron acceptor. They use just some other molecule, like nitrogen or sulfur or something like that. So you don't have to be aerobic to be alive. I think sometimes people make that leap in their mind, because there are things that don't really need oxygen. All right, so the first step is glycolysis, and people are like, yeah, can I learn glycolysis? And I'm like, I mean, sort of, but like not in the depth that you're thinking. If you were taking like biochemistry, you would have to realize that there are 6 intermediates on this arrow. And there's like a couple different enzymes in this process to get from glucose to pyruvic acid. But I don't make you know all the magic that's happening invisibly on the arrow. I just say glycolysis is the oxidation of glucose into pyruvic acid. So by oxidizing, you're stripping off the electrons, right? You're losing them in the form of a hydrogen. Glucose is a 6-carbon molecule. That's why it says 6C. You are oxidizing it with the help of different enzymes and converting it into 2 3-carbon molecules. Okay, so all you did was go from a 6-carbon to 2 3-carbon. So when you strip off those electrons, something is going to be there ready to accept it. And in this case of glycolysis, that molecule is something called NAD. So notice that nothing really gets lost, it just gets shifted around, because you had NAD, and now it becomes NADH2. Why? Because you stripped off 2 electrons. And by doing that, you have split the glucose into the pyruvic acid. Okay, so there's a bunch of different ways you can think of this, but if someone asks me, like, what's glycolysis? And these are the kind of things that are good, I think, study things. Like, don't just read what I said, like, try to make your own definition. Glycolysis is the oxidation of glucose into pyruvic acid. Like, that's what's happening in the image. So this process doesn't require oxygen. It's not oxygen dependent, so that's why most living things can do glycolysis. But if you're a complex organism like a human, glycolysis is not enough to sustain the energy that you need. And so you would die pretty quickly if you only were doing glycolysis. Like, it's just not enough energy. And the reason that is because it takes 2 to get started. So like maybe you have a funky little car, like where you're like, I gotta rev it up like to get it in the morning. And it's gonna go, but it still costs me a little bit of energy to get it started. And that's how glycolysis works, is that it's going to cost you 2 ATP to start it, and you're going to generate 4, so your net amount is 2. Okay, so you get some energy, but not like a ton at that point. All right, any questions about glycolysis? That's usually the first step of breaking down a sugar. Okay. Now, this is an aside to glycolysis. So imagine that we step over from glycolysis. We're not going down, but we're going like to the side. There's a process called fermentation. And fermentation is basically doing glycolysis in anaerobic conditions. And for reasons beyond what we really need to worry about, when you do things in the anaerobic conditions, it ends up making different byproducts. So it just drives it to a different chemical reaction. So humans can do fermentation kind of, but like not really. Like that's not really what we do. We do respiration. Okay, so respiration would be after glycolysis, you go to the Krebs cycle and the electron transport chain. And typically respiration is aerobic, but it can be anaerobic too for certain like bacteria. So a lot of microorganisms can do fermentation, like bacteria and fungus. And when there's less oxygen, they will do glycolysis, but instead of generating like carbon dioxide or going into the Krebs cycle, they'll actually make different byproducts, which are ethanol, which is alcohol, and gas in the form of CO2 actually. They do produce CO2 from this. So the example of that is like brewer's yeast is used to generate alcohol to make like wine and beer. And then the reason you use yeast when you're making bread is because it's going to ferment the sugars and produce gas as a byproduct, and that's going to make the bread rise. So we use it a lot for food industry stuff. Now, if you're a human and you're doing fermentation, let's say you're doing a very intense weightlifting type workout, or let's say you're doing a really fast sprint where you're not replacing the oxygen as fast as you're kind of using it, you temporarily go into this like kind of fermentative state, and humans will make lactic acid as a byproduct. And so that can cause problems with things like sepsis and stuff like that, like you'll see buildup of lactate and other things, but also that's what makes your muscles sore. Your muscle sore is the lactic acid buildup that came from that brief kind of fermentative reaction. So fermentation is kind of weird, I guess. Organisms can do it, but for some of them it's their main source of metabolism, but not for us. Like that wouldn't be our main way of getting energy, because you will get energy from fermentation, but it's not enough to sustain like human life, and it also has some kind of unwanted byproducts for us like lactic acid. But in microorganisms, we can use the byproducts to our advantage. Like we can see, we can use what they're doing as part of fermentation. Okay, so fermentation is what happens under anaerobic conditions. Glycolysis, it doesn't matter if there's oxygen or not, they can just do glycolysis. Like the cells are able to do that. All right, the last steps for organisms that do respiration is the cellular respiration part. You're continuing the ATP generating process. The molecules are continuing to get oxidized, so you're just stripping off the electrons, and when you strip them off, you have an acceptor waiting to collect that electron. And if you are an aerobic organism, like a human, then oxygen is the final electron acceptor at the end of the chain. If you are anaerobic, then it's just not oxygen. So it's something else like nitrogen or something like that. So a lot of things in life are aerobic, but there are certain microbes that are anaerobic, and then there's other levels to how anaerobic they are, which we'll talk about later. All right, so the next step after glycolysis is the Krebs cycle. In the older textbooks, they used to call this the citric acid cycle. So occasionally you might see an old diagram that says TCA, and that means citric acid cycle, like the citric acid cycle. But anyway, essentially what's happening is that you are continuing to oxidize the pyruvic acid that you generated in the first step, but before it can enter the Krebs cycle, you have to convert it into another molecule that's called acetyl-CoA. And acetyl-CoA is just a 2-carbon molecule. So see how it has 2 little circles? So at this point, you've gone from 1 6-carbon to 2 3-carbon, and now you're going to 2 2-carbon molecules. Okay, so nothing really got lost. You're just continuing to oxidize it. The way you oxidize pyruvic acid into acetyl-CoA is by removing carbon dioxide. And that's called decarboxylation. Like that's when you just remove a carbon dioxide. And this is why it's like a waste product for us humans. Like we just give it off as part of our process. So now most of the energy that was in the original glucose molecule is in the acetyl-CoA. Like acetyl-CoA is a very high energy compound. And now it will continue to get oxidized. And there's going to be, it's not showing it here, but there's enzymes, different enzymes that are doing each reaction. So notice that you do generate ATP from this by continuing to break down your acetyl-CoA. And then you have a new electron acceptor. So remember in the first step we had NAD, and now we have another one that's called FAD, and it's doing the same thing. So notice that FAD becomes FADH2. NAD became NADH2, and then eventually FADH2 and NADH2 are going to make their way to the electron transport chain so that they can become donors to start that process of oxidative phosphorylation. So if someone asks you like what the Krebs cycle was, you could be like, it's like the first step in cellular respiration. It's the oxidation of pyruvic acid into acetyl-CoA, and then the acetyl-CoA is further oxidized basically into electrons. So we've gone from, again to recap, 6 carbon to 2 3-carbon to 2 2-carbon, and eventually to like nothing. Like you just keep stripping it apart until it's elements basically. So when you do this, you end up getting 2 more ATP, and then the FAD and the NAD are now coenzymes for the electron transport chain, because they're going to help participate in that reaction by themselves getting oxidized, because they're going to give up their electrons and become donors, and that's how you're going to move through that system to make more ATP. Okay, any questions about the Krebs cycle? I don't know if it's something you can just read and absorb. Like, I encourage you to just like write it out, like, or draw pictures or whatever. Okay, so the electron transport chain, I have a bunch of text here that says what it is, but I like to just look at the picture because that makes more sense to me. But I put all the text there so that you would have it, okay? So essentially, what is it? It's a series of increasingly electronegative membrane-bound proteins that accept electrons in a series of redox reactions. So where is this happening? It depends on what kind of organism you are. If you are bacteria, it's just happening right in your plasma membrane, because they don't have organelles. If you're a plant, it's the mitochondria and the chloroplasts. The chloroplasts are where the photosynthetic reactions are happening. And then if you're an animal cell, then it's happening in the mitochondria. So they often will call the mitochondrion the powerhouse of the cell, because that's where you make most of your ATP, is in the mitochondria. So basically, the electrons that came from glycolysis and the Krebs cycle are taken by those coenzymes, which are the FAD and the NAD, and they're going to be given up to the first protein in the system, and then it's just going to be a series of redox, because when one thing gives it up, it's going to become oxidized, and the other thing's come reduced, and then it gives away its electron, and now it's oxidized, and the other thing is reduced. And it'll just keep traveling down that system until eventually it reaches a point where there's like a separation in charge across the membrane, and it forces the positively charged things that were repelled to come back inside, and it literally turns a crank inside of the enzyme. It's like the craziest thing that'll immediately start phosphorylating ADP into ATP. Okay, so you're turning it to that high energy molecule. And what continues this process, if you're an aerobic organism, is that there has to be an oxygen there to be the final electron acceptor. Otherwise, it's like shuts down that process. So I liked this drawing. So this is the one that I showed. Again, if you take biochemistry, you have to know a lot more. Like, there's probably energy associated with this, like to calculate, knowing what the different proteins are called, like in the order that they're in. But we just talk about it in a really general sense. So in the plasma membrane, or in the matrix of the mitochondrion, is where this is happening. So you did glycolysis, you did the Krebs cycle, and now all the energy that came from breaking those down is funneled into these electron carriers, which are your NAD and your FAD. So what they're going to do is offload, with the help of the enzymes, offload that electron, and then they end up getting oxidized. And then when the protein actually catches that electron, now it's reduced. And then it's going to give up its electrons, so the flow of electrons is here in orange. It gives it up, it's oxidized, the next thing is reduced. It gives it up, it's oxidized, the next thing is reduced. And then oxygen is hanging out there, if you're aerobic, to be the final electron acceptor. and you end up making water as a byproduct of that. Now, when the electrons move through, they are negatively charged, and so it forces positively charged hydrogens to repel out of the membrane. So now there's a separation in charge across the membrane. It's more positive on the outside and more negative on the inside. And remember that things want to flow with their concentration gradient. So said another way, now you have more hydrogens outside than you have inside, right? So they're going to naturally try to go back inside. And where they end up entering is in this enzyme here that's called ATP synthase. It has like an opening, a portal, that allows those protons, which are the hydrogens, to come in, and it turns that molecular crank. So you're rapidly going to convert ADP into ATP. So that part at the end is called chemiosmosis because it's a chemical gradient. Remember, osmosis was the movement of water across a membrane? This is a chemical movement across the membrane because the hydrogens are going across. And then this whole process of the redox reactions is called oxidative phosphorylation. That's the step where you're actually using those electrons and their transfer through a set of redox reactions to generate your ATP. So this process, I guess, is more complicated, but like not really. Like it's just different. And this is kind of where you get most of your cellular energy. Okay, it's going to come from the electron transport chain. Okay, so this is showing the summary flow. On the left side, they're showing the flow of electrons. On the right side, they're showing your ATP production. So remember, you go from glycolysis, you go from glucose to pyruvate or pyruvic acid, and you do that by stripping off electrons that get picked up by the acceptor. And then you take pyruvic acid and decarboxylate it, so you remove CO2, and then it can become acetyl-CoA. And then acetyl-CoA goes into the Krebs cycle, and you just keep oxidizing it, pulling off the electrons. And then when you get to the Krebs cycle, notice that a new coenzyme shows up, which is the FAD. And so we're not gonna balance it, you just take my word for it. But then eventually you have this many coenzymes that are carrying the electrons. And so then they come over to the electron transport chain and they pass their electron down that system. It causes that separation in charge, so it's going to fire up that enzyme, and then you're going to have a rapid conversion of ADP into ATP. If you're an aerobic organism, oxygen is going to catch the electron at the end. If you're anaerobic, it's not going to be oxygen. It's going to be some other molecule. So typically, if you were 100% efficient at doing this, you could get 38 ATP from one glucose. But do you always get exactly that? No, right? Because there's energy lost as heat. And sometimes you might not need to proceed all the way. So like what I mean by that is like, let's say you're like going along and you're doing glycolysis and Krebs cycle, and you're like, oh, I need to synthesize some lipids because whatever happened. You can take the acetyl-CoA and use it to make a lipid instead of using it to make ATP. Because a lipid can be made from acetyl-CoA, and a lipid can be broken down, or excuse me, let me say this again. Acetyl-CoA can be used to build a lipid, and a lipid can be broken down into acetyl-CoA. So that was the difference between catabolism and anabolism, is that the products that you generate during these steps can be used to build up on the anabolic side. And remember that building up will require energy, and that's why sometimes the energy will be lost. But it's not lost, it's just that you've used it for something else. So that's why you're not always going to get perfectly 38 when you first use that glucose. Okay, that's kind of a lot, but I think that's like one of the best ways I've ever explained it, if I do say so myself. Any questions on that? So there's a worksheet on all this that I think is helpful in the green book. Is it green? Yeah, the green one. All right, then I'm like, oh yeah, proteins. You can use those too for energy. So they would call this, I don't know what the term is, it's like, I'm having a brain fart. It's like amino acid oxidation, sort of. So basically, proteins are broken down into amino acids. Let me make this a little bit bigger. Okay, so amino acids are the building blocks of proteins. And so the enzymes that are going to break down a protein are different than the ones that are going to break down a sugar. The enzymes that break down proteins are generally called proteases. So you have intracellular protein, like you have protein that's just like inside of you, and then you have dietary protein. And so you convert that into keto acids and amino groups. And these aminos, like the NH, those can be used to build other stuff that's really important, like nucleotides, which is what your DNA is made out of, and then also amino acids, which are what other proteins are made out of. These keto acids can play as intermediates in the Krebs cycle. So it's like instead of starting with acetyl-CoA, if you have a protein, you're going to start somewhere in the middle of the Krebs cycle instead of at the top. Does that make sense? So it's just different starting material, so you can kind of jump in at a different spot. So a lot of times they'll say proteins are good energy sources because they're like slow burning. And I'm not going to get into like all the nutrition and metabolism stuff that has to do with that because I don't really know it. But it's like the difference between like, I guess carbs are like explosive energy kind of, and that's why you have that kind of crash afterwards. And then proteins, like the way you break it down is just differently, and so it takes longer to like process them. But notice that it still goes into the citric acid cycle on this diagram, which is the Krebs cycle. That's why I mentioned that. And then it'll go through the electron transport chain. And you give off CO2 and water as a byproduct. So to summarize that, protein catabolism can generate ATP. It can also provide the building blocks to make other proteins. And when you break down a protein, it can be converted into the intermediates that are in the Krebs cycle and the glycolysis. The intermediates are all the magical little things on the arrow that we didn't show you. So you know how it showed an arrow from point A to point B? The intermediates are the things that are on that arrow that we just don't need to know because of the scope of our class. All right, this is showing an example of lipid catabolism, which they also call fatty acid oxidation. If you look at the general skeleton of fat, it has a glycerol, and then it has these 3 fatty acids that are hanging off of it. So the fatty acid part can be used to make acetyl-CoA. And acetyl-CoA is a high energy molecule because that's what you use to start the Krebs cycle. And so fat is an energy source, but you also can store fat too if you don't need it necessarily for energy. And so this is just showing some biochemistry of fatty acid oxidation. And so as you oxidize that fatty acid, the fat, the lipid, the same kind of process is happening where you have electron acceptors that are there to receive the electrons that are removed as part of oxidation. And so you're just getting to different forms of it until eventually it chemically is acetyl-CoA. And that is the acetyl-CoA, the same one that starts the Krebs cycle. So hopefully you can see what I'm saying is like a lipid is not a sugar, right? So you can't do glycolysis on a lipid, but you can do fatty acid oxidation, separate these with an enzyme that's called lipase, and then use these fatty acids to synthesize through more oxidation acetyl-CoA, which is something that you can use for energy. Okay, any questions on the fatty acid oxidation, also known as lipid catabolism? Okay, so this is just another overview of the main metabolic pathways. So if you're an aerobic organism, you will go through glycolysis, the Krebs cycle, and electron transport chain with oxygen as the final electron acceptor. And typically you get 36 to 38 ATP. If you're an anaerobic organism, like bacteria, then you're going to do glycolysis and the Krebs cycle, but you're also going to use a non-oxygen compound as your final electron acceptor. So you use something like with sulfur or nitrogen or like carbon dioxide. And the energy seems to be way variable. I don't know why that is, but you can still make a lot of energy that way if you are set up that way, if you are an anaerobic organism. Then the last one is fermentation. So fermentation doesn't get to the respiration stage necessarily. So you start with glycolysis, but when there's not a lot of oxygen, then you're going to do fermentation using organic compounds as an electron acceptor, and then you're going to get alcohols and acids and gas as a byproduct. And so you don't make a lot of ATPs doing fermentation. So hopefully you can see with this visually, fermentation is not, it like skips the respiration step. Okay, it's not going to that end. Okay, this is showing the link between the three main nutrients and how they feed into deriving energy. So we spent most of our time talking about carbs. Carbs are broken down into simple sugars. Simple sugars, like glucose, can directly go into glycolysis. This is what I meant by intermediates, the thing on the arrows that you don't have to necessarily know. We just said glucose becomes pyruvic acid. And then during the Krebs cycle, after pyruvic acid becomes acetyl-CoA, then it'll become essentially electrons as it goes through the electron transport chain. And then those electrons will go through the chain. Oxygen is the possible acceptor, and then you make a bunch of ATP doing that. Now, if you start with a protein, you're not going to directly start at the beginning of glycolysis, because amino acids are used to make the intermediates along the way, and they're used to make acetyl-CoA, and they're used as intermediates for Krebs cycle. So do you see how it's a different starting point? Like imagine like you're running a race, and you started you and someone else started at the starting line, but imagine there's some dude like 2 miles down who was like, "Ooh, I'm starting now. Like I'm going to run in front of you." And to me, that's like kind of just, I guess the visual difference is like, I'm not starting quite at the beginning. I can start somewhere sort of in the middle if I take in protein instead. Same thing with a lipid, although lipids feed a little bit higher up in the process, but it's the same idea that when you take a lipid, you can break it into glycerol and fatty acid. The glycerol can feed into glycolysis. Fatty acid can directly be used to make acetyl-CoA. That's what I was showing you on that complicated diagram, that it went through like 4 or 5 different steps to become acetyl-CoA. All right. Now this is just mentioning the anabolic side a little bit. We've kind of joked before that you are what you eat. So whatever you eat is what you use to rebuild. The energy that's generated from your food can be used to build back up. So let's say you take in a starch, like a veggie or a potato, like carb, some kind of thing. You can convert that starch into glucose and then go through the steps to make ATP, right? But if you have enough glucose, you're like, I'll save this for later. And to do that, it's going to absorb energy because you are biosynthesizing a glycogen from a glucose. It's a molecule that has a bunch of glucose hooked onto each other. If you're a plant, you store sugar as starch. If you're an animal cell or a fungal cell, you store it as glycogen. Okay, let's say you start with a fat. You can break that fat down into what can become acetyl-CoA and then continue on your way to making ATP. Or fat becomes fat. So if you need to, store on your butt, which seems really like unnecessary, but you store it so fat can make other fat, right? Or you can use it for energy. Depends on what the system needs. Then if you take in a protein, you convert it into its building blocks, which are amino acids. Those amino acids can be used to make energy in the Krebs cycle, or you can use ATP and take those amino acids and make another protein. So that's why a lot of times, like when people are, working out and trying to build their muscles, they eat protein because you're tearing down your muscles and you need to rebuild them. And so having a good source of protein is a way to help you with that. Okay. This is just another 2 more diagrams that is explaining the difference between catabolism and anabolism. So when you take in food molecules, the energy is not easily usable, and so you're going to convert it into a useful form, which is the ATP. So that breakdown into the smaller parts is called catabolism. And you do lose some energy as heat that's given off, and that's why it's not 100% efficient. And now that you have these little building blocks, you can go through your anabolic pathways and input energy to rebuild back up whatever complex thing you need. Okay, so that's showing the link between catabolism and anabolism. Same thing here that we drew on the board at the beginning when we were trying to remember what we learned about. An anabolic reaction, your goal is to make a larger molecule, right? To do that, it costs you ATP, and you need the building blocks. So that is an anabolic reaction. So there's going to be enzymes that did this, that put this together. And then if you have a need for energy, then you can take a large molecule, like a protein, and you can break it down into its smaller parts by breaking the bonds. And when you break those bonds, it actually releases energy that you transform into ATP, like you store it as ATP. And now you have smaller molecules, and couldn't you use these to, go back over there? So hopefully you see the link between them. The starting material of one is the end product of another. The ATP generated from one process is used to begin the other process. All right. Okay. So I'm going to move on to chapter 6, which is totally different and easier, which is nice. So chapter 6 is very closely related to the lab. So it's talking about how do you grow these organisms. So if you want to culture them in the lab, what do they require? So we're going to talk about the physical and the chemical requirements for them to live happily in the lab. And then we're going to talk about different culture media. So this is one of the questions that you're asked on that getting to know a bacterium is how do you culture them in the lab? What kind of media is used? We'll talk about the growth curve, so how do they grow? And then a little bit about biosafety levels, which is how you work with them safely in the lab. All right, so we're dividing the requirements that these things have into 2 categories, which are physical and chemical requirements. So if you just think about yourself, for example, what kind of conditions do you need to be happy, or your best self? What kind of things do you personally like? What do we all need as humans? We need food and water. What else? Oxygen. Coffee. Comfy bed. Leggings. I'm never going to wear pants with a button again if I can help it. So we just need the things that make us comfortable, right? The things that keep us alive, we need those things. And microbes are not any different. They need those things too. They have their little bacterial leggings that they like. So they have things that they need to stay alive. So the physical things are kind of referring to literally the physical space, around, the environment that they need to be in. And so the main physical things that have to be met are pH, temperature, and pressure. Okay, so the physical requirements for growth are pH, temperature, and pressure. So like think about when we go on a plane, like they have to pressurize the cabin because our lungs will not inflate properly if the surrounding pressure is not correct. Okay. All right, so the first thing that we discuss is temperature. And basically you can categorize the microbes based on the temperature that they grow the best at. So where they grow the best is called their optimal temperature. But what you'll notice is that there is a range of temperatures that they could technically grow. Anything outside of that range, they're not going to be growing very well. And that's why you see it like go off. So the categories are based off of where their favorite temperature is. So if they call them a psychrophile, psychro means cold. So these are cold-loving critters. So this one's actually called polar amonas. I'm assuming that they found it at the pole, like they found it at like the North Pole or the South Pole or whatever. And it's growing around 4 degrees Celsius, which is just above freezing. Freezing is 0. So this likes to grow the best at 4. So that's a pretty good example of a psychrophile. Most human pathogens are mesophiles. So meso means moderate or middle. They like temperatures that are kind of around our body temperature. Like humans have a very pleasant body temperature. And that's why a lot of the pathogens will thrive there. And so human body temperature is 37 degrees Celsius. So E. coli likes to be right around that, a little bit higher. But notice that E. coli has a very wide range that it can grow. And that kind of has implications for it as a pathogen, right? Because the wider the temperature you can adapt to, then the more, like the better of a pathogen you could be. If you only grow exactly at 39 degrees Celsius, what's going to happen if you're not at 39 degrees Celsius? You're going to die, right? Or you're not going to grow. So it is an advantage for a pathogen to have a range of growth temperatures. Then you have things that like it a little bit hotter than that, not quite boiling, somewhere between human body temperature and boiling. So boiling is 100 degrees Celsius. So things that are sort of in the middle of 37 degrees Celsius and maybe 100, they call those thermophiles. So some of those were discovered in like some of the springs, like hot water. But the ones that are in boiling almost conditions are called hyperthermophiles. So those are going to be the archaea. So a lot of people were like, I forgot what archaea is. Archaea are prokaryotic extremophiles. So they're going to be like in super freezing or super hot or super acidic or super basic conditions. So the 3 main categories are psychrophile, mesophile, and thermophile. And most human pathogens are mesophiles. So as I mentioned, every organism has an optimum or optimal temperature. That's where its metabolism is at its highest rate. So if you're a mesophile, that would be around maybe human body temperature. If you're a psychrophile, it's going to be maybe in the refrigerator. Like listeria can grow in the fridge, and that's why we see outbreaks. Like we saw the outbreak with listeria and ice cream, the Bluebell ice cream a while back. Because listeria is a psychrophile. It loves refrigeration, so it'll just hang out in there and grow. And then thermophiles need it to be a little bit warmer. So whatever the bacterium wants, where it's going to grow the best, that's called the optimum. And then every bacteria has a range of temperatures that it can grow at. Some of them have a really narrow range. Some of them have a bigger range. The minimum temperature is the coldest that they could survive. And really, they literally solidify. Like the plasma membrane will start to gel up. And then that's going to slow down transport and metabolism for the bacteria. On the opposite end, if they get to the maximum temperature that they can stand, notice that they're not really growing either. They're like on that verge of death or stopping their growth. When you get too hot, you denature the proteins. And you literally collapse. Like the cell will lyse due to thermal pressure, like from the heat. So it's not good to be at the extremes of temperature for bacteria because they stop growing or they die for different reasons. When it gets too cold, metabolism slows down. When it gets too hot, the cells literally explode. Like they'll just pop. So there is, of course, then, if you think about food safety purposes, there's kind of this danger zone that's between 20 and 50 degrees Celsius. 20 degrees Celsius is right around room temperature. And that's why it's important to refrigerate your leftovers promptly. Because if you keep your food in this danger zone, bacteria are going to be growing potentially very happily. And that's why it's important that you either cool it down to store it, or when you're heating it to cook it, they each have a minimum temperature that they're supposed to reach. So my husband and I were actually chatting about this the other day. I guess a couple years ago, the USDA lowered the temperature that they said was okay to cook pork to. So my husband loves to grill and smoke and stuff, and I think the temperature requirement for pork was 165 degrees Fahrenheit. For whatever reason, they lowered it to 145, which I don't think that'll make pink pork, but it's just we were nah. We had it so ingrained in our head that pork should not, be underdone because of the concern with tapeworms. But I wonder if they lowered it because we literally don't really have tapeworms in the U.S. And so maybe that's why they were you don't have to heat it as high as we thought. But you do you. I'm still doing the 165. Okay, the other physical requirement is pH. So we just talked about temperature. Now we got pH. And pH is how acidic or basic or alkaline a solution is. The way I always remember pH in my head is pH is potential hydrogen. So it's what is your balance between acid and base? What they're calling acid is your H plus ions, and then the base are your OH negative ions, the hydroxide versus hydrogen. When you're right in the middle, that's when OH is equal to H. So the acid and base is balanced, and that's called neutral. And that's 7.0 on this scale. Anything below 6.5 in the middle would be considered acidic. It's going to have an excess amount of hydrogen ions. Anything that's above 7.5 is considered basic, and then that's going to have excessive OH. Okay, so notice that a lot of the foods that we just eat are kind of at neutral pH, and that's why a lot of foodborne outbreaks happen with those foods like meat and cheese to a degree, and milk and water, because it has a pH that can sustain bacteria, potentially, that could cause disease in humans. So remember that the significance of your pH being too high or too low is that you denature the proteins. And if you denature the proteins, then you either lose enzyme function, which is going to slow down metabolism, or you can also denature the structural proteins that are holding the cell together. So most things in life prefer a neutral pH, but some things can survive at lower pH, like fungus. Some things actually like acidic pH, those are called acidophiles. And some things actually like more base, and that's called alkalophiles. So you do have the extreme ends, but again, most things like to be at neutral pH because it's not burning you when you're at neutral pH. Right, the last physical requirement that we consider is pressure. And a lot of times, not only is this atmospheric pressure, like the surrounding pressure, but it's the pressure that's generated from osmosis. So the cells are surrounded in water, and water can move freely across the membrane. And when it does that, it exerts a pressure on the cell. If there's an equilibrium of flow, then the cell will be okay, it'll be in that isotonic state. But if you put it in a salty solution where there's less water, water is going to leave the cell, and it could potentially rupture. And that's kind of what it's showing here, is that when they put a bunch of salt water around a plant cell, the cytoplasm is actually pulling away from the plasma membrane, and that cell will eventually like rupture and die. On the flip side, we can use this to our advantage. A lot of food preservatives are salty. Like, remember when they settled the Old West, they're like, oh, we got all this meat, let's salt it, like let's cure the meat. And by doing that, you're drying out any contaminants that are in there so that they can't spoil your food or make you sick. So when we would dilute our bacteria in the lab to plate it, we would use a substance that was called phosphate buffered saline. It's just a saline solution. Because if you use just pure water, it's going to rush into the cells and pop them. So then they would be dead and we wouldn't see them on the plate. So we had to buffer it with salt to make sure that the osmotic pressure stayed stable. Now there are some things that like salt, and those are called halophiles. Most things are facultative halophiles, which means that they can grow with or without salty conditions. So what is one thing that you can think of that grows on your body that likes salt, probably? A bacterium. What part of your body is salty besides your spirit? Salty spirit. What other parts of your body are salty? Your skin, right? So what grows on your skin? Staph, right? Like staph is known to just do really well on skin, and it's because it's a halophile. It likes salt. So then related to that, what if you wanted to grow staph in the lab? How could you get rid of the things that you're not interested in? You add salt to the media. And staph is like, yay, salt. And everything else is like, oh no, salt. And that's how you isolate the thing that you're interested in. That's called selective media. So there is a selective media for staph that's called mannitol salt agar. It uses salt to select for your staph. And we'll use that towards the end of the semester. Okay. Then there's the chemical requirements. And basically the chemical requirements are like food. Okay, so chemical requirements is like food that you need, atoms, or I shouldn't say atoms, I'm going to say elements, vitamins, and minerals, which are often part of the organic molecules that you need. So those are things that you might not be able to make yourself. You just need to get them from your diet. So chemical requirements are food. Food, elements like carbon, nitrogen, phosphorus, sulfur, potassium, minerals, those all have a function in the cell to keep it alive. So elements are one part of your chemical requirements, and everything needs carbon. So I would even make a note to say carbon. Okay, we're carbon-based life forms. We have to have carbon as an energy source sometimes, but we're literally made out of carbon. Like all the organic molecules are made out of carbon. Okay, the other chemical requirement besides carbon is oxygen. But I don't mean oxygen to build up something. I mean, do you need oxygen for metabolism? So not all things do. And usually how you're kind of tested to know these categories is that if you look at the terms in blue, that's the oxygen requirement, and that correlates with where they're growing in the test tube. Another thing is that oxygen at high levels is actually toxic. And so if you are anaerobic, you don't have the enzymes to reduce the toxicity of oxygen. So if you look at where they're growing in the tube, that's how you know what they are. So the first tube is an obligate aerobe. It needs oxygen. It's obligated to be aerobic. How do you know that? Because it's growing at the top of the tube, where there's more oxygen. So which one is the opposite of this? All the other ones that say various anaerobes, right? But this one is the straight opposite. This is an obligate anaerobe. Does not like oxygen, it's hiding in the bottom. Most human pathogens, a lot of human pathogens, are facultative anaerobes. They can grow with or without oxygen. They prefer oxygen, because notice that more of them are at the top, but they can get by with less oxygen. Another flexible one is this one here, which is called an aerotolerant anaerobe. It tolerates air, but it still is anaerobic. It would prefer not to have air, but it can tolerate it. So which one do you think is easier to grow? That one or that one in the lab?
D.
Yeah, right? Because you have to remove oxygen from the atmosphere. You have to put it in the incubator if you want to grow a strict anaerobe, an obligate anaerobe. The last one is the pickiest one, and the term for picky is fastigious. Those are the ones that have a lot of growth requirements to coax them into growing. So these ones are called microaerophile. Micro means a little bit, aero means oxygen. They love exactly a little bit of oxygen. So if you remove too much oxygen, they will die. And if you give them too much oxygen, they will die. So notice that they're hanging out in the middle, okay, in that sort of state where the oxygen is not too high or not too low. And a lot of them like other gases like CO2 as part of incubating them. So to understand the oxygen requirements, the categories, it helps to look at the tube. They'll grow where the oxygen suits their needs. All right, so I'm going to stop there. Sorry I went over a minute. We'll continue with this next time and then move into chapter 7. Okay, I'll see you guys in lab. We'll start lab at 11:30, so we have a little break.